Hi friends, my name is Nandita. I am from Andhra Pradesh.
Good morning dear students. Today we are going to discuss about locomotion and movement from chapter 20 biology class 11 CBSE syllabus. Children, let us discuss about structure of muscle. Each organized skeletal muscle is made up of a number of muscle bundles or fascicles held together by a common collagenous connective tissue layer called fascia. Muscle is composed of a large number of elongated cells called muscle fiber. The outermost covering of muscle is called epimysium. Each bundle of muscle fiber called fasciculus is surrounded by another connective tissue covering called perimysium. Inside fasciculus, there are several muscle fibers, each surrounded by connective tissue covering called endomysium. Each muscle fiber is lined by the plasma membrane called cytoplasma, enclosing by cytoplasm. Children, let us discuss about muscle fiber and sarcomere anatomy. Skeletal muscle are present in the animal body most abundant. They are composed of bundles of muscle fibers surrounded by connective tissue. Fascicles, a muscle such as a biceps, is made up of numerous muscle bundles that are held together by fascia. A connective tissue layer and several muscle fibers are found in each fascicle. Children, let us discuss about muscle fibers. Muscle fibers are long cells. They are grouped into fascicles. Muscle fibers have the following characters like skeletal muscle fibers are all long, cylindrical and serrated. It is syncytium, which means it has many nuclei. The plasma membrane of the muscle fiber is known as the sarcolemma. Sarcoplasma is the muscle fiber's cytoplasm. Muscle fiber is syncytium, as the sarcoplasm contains many nuclei. Endoplasmic reticulum, that is sarcoplasmic reticulum of the muscle fibers, is the storehouse of calcium ions. A characteristic feature of the muscle fiber is the presence of a large number of parallelly arranged filaments in the sarcoplasm called myofilaments or myofibrils as a contractile element. Children, let us discuss about myofibrils. Multiple myofibrils run lengthwise and adjacent to each other in the sarcoplasm of each muscle fiber. Myofibrils alternating dark and light bands bring the muscle a straight look. Each myofibril is made up of even simplified structures known as myofilaments. Each myofibril has alternate dark and light bands on it. The light bands contain actin and it is called I band or isotropic band, whereas the dark band called A or anisotropic band and contains myosin. Both the proteins are arranged as a rod-like structures parallel to each other and also to the longitudinal axis on the myofibrils. Children, let us discuss about myofilaments. In a myofibril, there are two kinds of myofilaments. One is thin filament and other that are thick. Muscle contraction requires the attachment of thin and thick filaments. Now let us discuss about actin filaments or thin myofilaments. Actin filaments are thinner as compared to the myosin filaments, hence are commonly called thin and thick filaments respectively. The thin filaments, also known as actin filaments, are composed of three types of proteins. It is made up of two polymeric filamentous or F-actin filament that are wound around each other. They are globular or G-actin monomer polymers. Tropomyosin filament coil lengthwise around actin filaments. Troponin, which is found at specific points on tropomyosin, protects the active binding zones for myosin. Troponin and tropomyosin govern actin and myosin filaments binding and those play a significant role in a muscular contractions. Children, let us discuss about myosin filament, which is also called thick myofilaments. Myosin is used to make thick filaments or myosin filaments. It is a polymeric protein made up of polymeric units of the protein meromyosin. It is composed of three parts: a tail, a short arm or neck, and a globular head. At regular times, the head and across arms extends from the filament. The globular head compresses ATP and act binding sites. The enzyme ATPase is activated by the head. Children, let us discuss about sarcomere. A sarcomere is a muscle contraction functional unit. A myofibril is made up of hundreds of sarcomeres that are linked end to end. Sarcomeres are the fundamental unit of muscle contraction. It is made up of actin and myosin filaments repeating units in a particular order. Sarcomeres are merged at the ends by filaments that interweave to form the Z line. It is a sarcomere limiting membrane. At the regular intervals, actin or thin filaments are linked to the Z line. Between the thin filaments or thick or myosin filaments, they are kept together by a really thin fibrous membrane called the M line. Actin and myosin filaments span each other in a particular pattern, resulting in a muscle striation. They form three kinds of bands. The I band or isotropic band is the light band, and it is made up of actin filaments connecting by two adjacent sarcomeres. Thick and thin filaments do not overlap in this case. The A band or anisotropic band is the dark band which contains both actin and myosin filaments that overlap. The H zone is the myosin filament center portion where actin filaments do not coincide with thick myosin filaments. In the center of each I band is an elastic fiber called Z line which bisects it. The thin filaments are firmly attached to the Z line. The thick filaments in the A band are also held together in the middle of this by a thin fibrous membrane called M line. The A and I bands are arranged alternatively throughout the length of the myofibrils. The portion of the myofibril between two successive Z lines is considered as the functional unit of contraction, and it is called a sarcomere. Hence, sarcomere are the contractical units of myofibrils. In a resting state, the edges of the thin filaments on either side of the thick filaments partially overlap on free ends of the thick filaments, leaving the central part of the thick filaments. This central part of the thick filament not overlapped by thin filaments, and it is called the H or Z bands. Children, let us discuss the structure of contractical protein. The muscle fibers are composed of 20% protein, 75% water, and remaining 5% is composed of inorganic and organic matters. Muscle proteins are myosin, actin, tropomyosin, and troponin. Myosin. Thick filaments are made up of myosin molecules. Each myosin molecule contains two identical heads with a pair of helical strands which forms the tail. The upper surface of each globular head contains actin binding site and ATP binding site. Actin. It is the main constituent of thin filament. The actin filament resembles two strings of beads twisted in a double helix. Each bed is a molecule of J-actin having 55A diameter. It shows high efficiency for calcium ions. In the presence of salts and ATP, it is converted into fibrous actin, F-actin. Tropomyosin. It is a two-stranded helical rod which is located in the groove between the two helical strands of actin. A troponin complex is attached to the tropomyosin at regular intervals of about 385 angstroms. Troponin. Troponin is a globular protein. It consists of three polypeptide chains and the calcium binding subunit, the inhibitory subunit, and the tropomyosin binding subunit. It is an important control protein. It has high efficiency for calcium, which calcium binds with the troponin. The active sites of active protein are exposed. Children, let us discuss about muscle contraction mechanism. Mechanism of muscle contraction is explained by Seidling filaments theory, which states the contraction of a muscle fiber takes place by the sliding of the thin filaments over the thick filaments. Sliding filament theory of a muscle contraction was proposed by A.F. Huxley and H.E. Huxley in 1954. According to this theory, during muscle contraction, actin filaments slide inward on the myosin filament of A band with the help of cross bridges to reduce the length of the sarcomere. Sliding increases overlap of a thick myosin and a thin actin filaments and caused the sarcomere to shorten and the muscles contract as the result of this. The brain or spinal cord sends a signal to motor neurons to initiate muscle contraction. The neural signals induces the neurotransmitter acetylcholine to be released at the neuromuscular junctions. Sympathetic acetylcholine binds to receptors on muscle fibers, causing sarcolemma depolarization. The generated action potential propagates through the muscle fibers. The sarcoplasmic reticulum releases Ca2 plus ions into sarcoplasm. Another protein, dystrophin, regulates Ca plus 2 release. Ca plus 2 ions interact with troponin and alter its conformation. Myosin binding areas on actin filaments become visible. The myosin head also has an ATP binding site where ATP binds. The myosin head's ATPase activate catalyze ATP hydrolysis. They coact myosin heads binds to actin, activate binding sites, creating a cross bridge. Following the attachment, the myosin head releases phosphate, resulting in the power stroke. Myosin filaments deforms and pulls actin filaments towards the sarcomere center, causing the sarcomere and the muscle to shorten. During this process, ADP is released and ADP also responsible for myosin head detachment. This process is repeating in the existence of sufficient Ca plus 2 ions. Children, let us discuss what is muscle relaxation. When a neural signal stops, acetylcholinesterase is a protein that deactivates acetylcholine in the sympathetic cleft, which fibers enter a state of rest. Ca plus 2 ions are returned to the sarcoplasmic reticulum. In the complete lack of Ca plus 2 ions, the troponin and tropomyosin complexes recovers the actin filaments, myosin bind sites, and the sarcomere Z lines come back to its original position and muscle relax. Children, let us discuss about muscle fatigue. ATP derives muscle contraction and ATP is obtained by muscle fibers from the backup of creatine, phosphate and glycogen. Under normal conditions, glycogen is turned into glucose, which is then used to produce ATP during cellular respiration. Muscles require a lot of energy when doing strenuous exercise. The body could not substitute the oxygen demand and glucose is degraded anaerobically. As a result, lactic acid builds up, causing muscle fatigue. Rigor mortis or short-term muscle stiffening after death is caused by ATP depletion as cellular respiration terminates. ATP is necessary for the dissociation of myosin head. In the absence of ATP, the cross bridges in the muscles that were contracting remain intact. It aids in calculating death time. Children, let us discuss about major types of skeletal muscle fibers. Muscle fibers are classified into two types based on the quantity of oxygen binding pigment, which is myoglobin, present in them. Based on the sarcoplasmic content, situation of nuclei and a number of formed elements, two types of fibers are recognized in a striated muscle, that are white and red fibers. White fibers, anaerobic muscles, are pale or white and have less myoglobin with mitochondria but much more sarcoplasmic reticulum. White muscle fibers are fat muscle fibers which lack myoglobin and have fewer mitochondria. These muscle fibers perform fast work for short duration, as the result of which these fibers get fatigued quickly. Example, eyeball muscles flight muscles of sparrow. Now let us discuss about red fibers. Red fibers are also known as aerobic muscles, are reddish and contain too much of myoglobin and mitochondria. Red muscles are slow muscles fiber processing red heme protein and abundant mitochondria. These muscle fibers can perform slow sustained work over a long period without getting fatigued. Example, extensor muscle of back and flight muscles in kites.